Hello everyone. In this video, we'll talk about Chargaff rule, what Chargaff did and what Chargaff concluded, what were his major findings, what was his rule. We'll discuss in this lecture, in this video lecture. So Chargaff rule and conclusions, they are all about DNA molecule, double-stranded DNA molecule. So in late 1940s, Edwin Chargaff collected DNAs of many organisms from same species and different species, and he performed some experiment and concluded some findings. Now these findings, they are called as Chargaff rules. Now to understand these findings, this rule, you should know some basic but important points, which are key points. Now we'll see these points one by one. We'll get these points one by one on this page. So number 1 point is that many of us know that in each organism, each cell has a DNA molecule. Now we know in our organism, now there are many tissues and many cells. Now these cells, they have their own DNA molecule. Second point is that this DNA molecule, it is double stranded, means it is duplex, means what? This DNA molecule, it contains 2 strands. Now number 3rd point is that these 2 strands of DNA, they are complementary to each other in nitrogenous base pairing. Now these 2 strands of DNA which is there in a cell, they are complementary to each other in nitrogenous base pairing. Now you know nitrogenous base pairing. Now you should remember that in a DNA molecule there are 4 nitrogen bases. Number 1, it is adenine, which is denoted as capital A. Number 2, it is guanine, which is denoted as capital G. Number 3rd, it is cytosine, which is denoted as capital C. Number 4th, it is thymine, which is denoted as capital T. So remember that out of these 4 nitrogenous bases, adenine and guanine, they are purines, and cytosine and thymine, they are pyrimidines. Now next point which you should remember is that in 2 strands of DNA, adenine always pairs with thymine with 2 hydrogen bonds, and cytosine, it always pairs with guanine with 3 hydrogen bonds. A, it pairs with T, and C, it pairs with G. Now you can see this in a figure which is there on your screen. So A, it is pairing with T. Now A is on one strand and T is on another strand. So A is pairing with T, G is pairing with C. Now look at the hydrogen bonding. A is pairing with T with 2 hydrogen bonds, and G, it is pairing with C with 3 hydrogen bonds. So now we'll see what exactly is the Chargaff rule, or what does Chargaff rule state. Now Chargaff rule, it states that in a DNA molecule of any species, the ratio of purines to pyrimidine, it is always 1:1. Now what he is telling, now what this rule is saying, now it is telling that the ratio of purines to pyrimidine, it should be 1:1. Now let's understand this statement, this rule, by taking an example of DNA molecule. So look at the DNA molecule which is there on your screen. Now look at the nitrogenous base pairing and count the number of purines and pyrimidines. Now you know purines are adenine and guanine, and pyrimidines are cytosine and thymine. Now count the number of cytosine and thymine. Cytosine is 5 in number and thymine is 6 in number. Now adenine, it is 6 in number, and guanine, it is 5 in number. Now look at the figure. Now adenine number 1, 2, 3, 4 and 5. Likewise you can count the number of purines and pyrimidine. So number of purines, total number of purines, it should be A plus 2, because purines are adenine and guanine. So total A plus G, it is equal to 6 plus 5 is equal to 11. And number of pyrimidines, total number of pyrimidines C plus T, because pyrimidines are cytosine and thymine. So 5 plus 6, it comes 11. So what will be the ratio of purines to pyrimidines? It will be 100% purines A plus G by pyrimidines C plus T. Now look at the screen. Number of purines 11, A plus G 11, and number of pyrimidines C plus T, it is 11. 11 by 11, it is 1 by 1. So therefore ratio is 1:1. So this is correct. What Chargaff rule states, it states that purines to pyrimidine ratio in a given DNA, in every DNA, it should be 1:1. So we have proved that rule, and now we have understand this rule. Now some statements are there which depends on this Chargaff rule, and these statements, they are very important regarding competitive examination like NET, SET, GET and other examinations. So number one statement is that number of adenine is always equals to number of thymine. 100%. It is obvious because adenine, it always pairs with thymine. So number of adenine, it should be same with number of thymine. So A, capital A, it always equal to T numbers. Now second statement is same, number of cytosine is always equals to number of guanine. Obvious. It is also obvious because DNA strands, they are complementary and their pairing is specific. Cytosine, it always pairs with guanine. So if cytosine is 3, guanine will be 3. If cytosine is 6, guanine will be 6. If cytosine is 7, guanine will be 7. So their number will be same. So C, number of C is always equal to number of G. Now therefore, now look at this screen. Now A and T, they are equal. So A plus T, their addition is always equal to C plus G, 100%. Now take an example of our DNA molecule A plus T by C plus G, because equal, they are same. So same by same is equal to 1. Their result should be 1. Now third statement is that adenine percentage is always equal to thymine percentage, and cytosine percentage is always equals to guanine percentage. Now this is important regarding NET SET examination. Now what is the statement? Percent A is equal to percent T and percent C is equal to percent G. Now these four nitrogenous bases A, T, C, G, they make 100% of DNA. So out of this 100%, A always equal to T, percent A is equal to percent T and percent C is equal to percent G. So number fourth point is that ratio, now look at the point, ratio of adenine plus thymine by cytosine plus guanine is not equal in different organism, but this ratio is same in same organism but in different tissue. Now in one organism there are many tissues and cell, so in that organism this ratio A plus T by C plus G, it is same, but it differs in organism. So take an example of our own DNA, previous DNA. So adenine is 6, thymine is 6, cytosine is 5, guanine is 5. So A plus T is equal to 6 plus 6, it comes 12, and C plus and cytosine, it is 5, guanine, it is 5 in our DNA molecule, which we the example we took in previous pages. So G is 5, so C plus G, 5 plus 5, it is 10. Therefore A plus T by C plus G ratio is 12 by 10. Now 12 by 10, it comes 6 by 5. So ratio is 6 by 5. 6 by, as to 5 means this ratio, what that statement is saying, this ratio, it changes in different organism, but it is same in it is same in different tissues of same organism. Now look at the numerical problem of Chargaff rule. Now it is very important. Now question is that a segment of DNA has 120 adenine and 120 cytosine bases. The total number of nucleotides present in the segment is, now you have been asked to count the total number of nucleotides, not the nitrogenous bases. Remember the difference. Now what will be the answer? Now to see complete solution and other questions related to Chargaff rule, please watch my next video. Please subscribe, like and comment to my channel. Thank you.